Welcome everybody to our May EpIC seminar. Just a quick reminder, this is space primarily for early career researchers and senior postdocs to tell us about their work in electrophysiology and ion channels. If you know somebody whom you think would be great to see here, please do use our webpage. You can nominate people, you can also self-nominate, and we do take all these nominations into consideration, and we already have a few speakers from your nominations, essentially. So our May speaker is Claudia Moreno. Claudia did her PhD in Mexico and then went on to do a postdoc in Fernando Santana's lab at the University of Washington. This is where she discovered a new regulatory mechanism of L-type calcium channels via their C-termini. And after that, she got a K99 award towards independence from National Institute of Aging at UC Davis. And in 2019, I think she started her group at University of Washington again, still working with calcium channels and their role in aging. She's now there also a Freeman Hrabowski Scholar who recognizes, this is an award that recognizes new early career researchers with a great breakthrough potential. So I'm very excited to hear Claudia's latest work. Over to you, Claudia.
Thank you so much. Let me share my screen first. Okay. Are you seeing my screen now?
Yes, we can see it.
Yeah.
Okay, display. Perfect. Thank you so much for the introduction, Helena. It's a pleasure for me to be here in the EpIC seminar series. Thank you so much, Connor and Osama and Laila also, who's not here, but for considering me for this really nice space that I think is nurturing the next generation of ion channel lovers. So I'm really glad that people are attending this seminar. Thank you for those in the East Coast that are taking your lunch hour to be here with me, and those in the West Coast that are probably dedicating your more productive hour to be here in this seminar. So today I'm gonna share with you some of the research that we are doing in my lab at the University of Washington on ion channels and aging. So that's the general title, which will change it back and forth, but I decided to stick with the first one because I'm gonna talk mainly about ion channels and aging. So the main goal of the talk today is that you all leave with a better idea of three things. The first one is how the cardiac pacemaker makes our heart beat. The second one is the important role that voltage-gated calcium channels play in this mechanism. And the third one is how aging messes up with this channel. So the vision, so before I jump into the research, I want to acknowledge the people from my lab that have participated in this project, specifically some former members on your screen: Matias Baudot, Sabrina Shoy, Viviana Vargas, and Gabriela Serrano contributed a lot to the data that I'm going to show today. And also some people from my current group, Roxanne Madden, and people from Oscar Vivas, a group that he's my close collaborator in this project. Okay, so the vision of my lab is to understand the mechanisms behind heart rate adaptability. And we studied this at different time scales. From short-term adaptability, the one that occurs in the order of seconds or hours, like the instantaneous change in heart rate that we experience when we sprint, or when we are taking a test or giving a seminar, to also daily changes in heart rate observed during our awake and sleep cycle. Middle-term adaptability that concerns changes in heart rate happening in the order of years. For example, those changes occurring along the lifespan of an individual, like the ones we're going to talk about today, that are what happens with heart rate as we age. To long-term adaptability, in which we are more concerned about studying the adaptations that animals have evolved to sustain the broad range of heart rates that are exhibited in nature. Today, we're going to focus mainly on heart rate adaptability at the middle term that is related to the aging process. So, but let's understand first how heart rate is produced and controlled. And here's where we're gonna dive into the pacemaker. So probably if we ask people around the world, many people are familiar with that word, pacemaker, the cardiac pacemaker. But if you ask people to select an image that corresponds to what a cardiac pacemaker is, probably most of the people would select option B. So here's the first main goal of the talk is that all of you live with a visual idea of how the real biological pacemaker looks like. And so that's option A. For those of you that have never seen a pacemaker, this is how the mouse pacemaker looks like. As you can see in the image, this is a cleared mouse pacemaker. So it's the intact explant that we extracted from the heart. And we immunostain this against the HCM4 channel, that is one of the main markers for pacemaker cells. And you see how this labeling shows the very elaborated architecture that the pacemaker has. So in the upper part of your, just let me see if you see that red dot that I'm using as a pointer. Yes, okay. So in the upper part, you see here, this is what we call the head of the pacemaker. And then we have another part here that is called the body of the pacemaker. The pacemaker is a very conserved structure among vertebrates. It looks very similar and is located in the same anatomical place in the heart. So to give you more context on where exactly is this in the heart, for those that are not that familiar with the heart. So you see here a diagram of the heart, and that yellow region that is depicted there in the diagram is where the pacemaker is located. So the pacemaker is located at the sinoatrial node that is right next to the right atrium. So this small and specialized tissue, as I mentioned, is very concerned between among vertebrates, and it's formed by electrically coupled cells that generate and propagate a rhythmic signal that travels through the tissue in the form of a calcium wave. So calcium plays, therefore, a very important role in conducting electricity across the heart. So each of these cells that form the cardiac pacemaker is an individual oscillator. And so what you are seeing now on the right of your screen is a pacemaker cell that we isolated from this tissue. And so you see that they look very different from what you have in mind about how a cardiomyocyte looks like. And the reason is because they functionally are very different. These cells are not, their main purpose is not contraction as the rest of the cardiac muscles, but these are cells that their main function is generating rhythmic electrical signals that trigger the heart contraction. So inside these cells, obviously, to achieve this automaticity, they have a set of specific ion channels that underlie the pacemaker action potential, and we're going to see this in more detail. So this electricity that is generated by the pacemaker travels along what is known as the cardiac conduction system, that is the rest of the yellow wiring that you are seeing on the heart. And the conduction of this electricity is what triggers the depolarization of the cardiac muscle and therefore triggers the contraction. So this cycle is repeated over and over again since the moment the pacemaker is formed in the embryo until the very moment we die. So it's a very robust system. And for example, in humans, this cycle is repeated 100,000 times a day and near 2.5 billion times in an average life. So how fast our heart beats then depend on the intrinsic heart rate intrinsic firing rate of this pacemaker cell. That heart rate is also controlled by the nervous system, because as you can imagine, there are moments where we need our heart to beat faster or slower depending on the physiological demands. So for this, the pacemaker is very highly innervated by the autonomous nervous system, and it receives terminals from both the sympathetic, shown there in magenta, and the parasympathetic, shown in green branches. So these two branches work as the accelerator, pedal, and brake of the heart. So when we need our heart to beat faster, the sympathetic branch is activated, and through the release of noradrenaline acting directly on the pacemaker cells, the firing of these cells is accelerated, and then our heart beats fast. On the other side, when we need to slow down, that many of us need that very often. So that's what is behind all the breathing exercises that people now practice, and mindfulness is trying to activate the parasympathetic branch. And now through the release of acetylcholine acting again on the pacemaker cells, the firing rate is decreased, and now the heartbeat is low. So this is a very robust system, as I said, but unfortunately is not exempt of the deleterious effects of aging. So aging, as you can see here in this graph, causes a linear decline on the maximum heart rate and also in the intrinsic pacemaker rate, as is shown in orange. So this happens in all mammals, and something that is very important, I'm gonna go back to this in the second part of the talk, is that you can see here there are two components. So we have what is called the intrinsic pacemaker rate in orange. That is what I mentioned before. It is the rate at which your pacemaker cells beat if we take them out of your body and out of the heart without any neuronal input. So that's the spontaneous firing. And then in green, we have the resting heart rate in the case of humans that we have a tonic parasympathetic inhibition, and we see that basically that doesn't change through our life. And in maximum heart rate here in magenta, it also follows the same trend than the orange trace, with a linear decline with age, and that's that decrease in maximum heart rate is one of the main reasons because it becomes so difficult for us to perform aerobic activities when we get old. So in humans, this slowdown can become pathological. And in fact, the slowdown of the pacemaker is the main cause for the requirement of the implantation of artificial pacemakers, as you can see there. And this is the cause for people requiring the option B, that is the artificial pacemaker. So artificial pacemakers implants surpasses in the US 200,000 implants per year, and it's a problem that obviously with the increase in the life expectancy is becoming more prevalent in humans. So then the main question here is why the pacemaker slows down as we age. And this is not completely understood. And this research line of my lab focuses in identifying molecular mechanisms behind this age-associated slowdown of the intrinsic pacemaker rate. So when we talk about molecular mechanisms, then we have to go into the pacemaker cell and see what are the molecules that make these cells fire. So we're going to use this diagram to highlight some of the main components, because now obviously I have an audience full of ion channel lovers with their favorite ion channel in mind at this moment, hoping for that channel to jump in the screen. So just don't get mad if it doesn't appear. I have to just mention the ones that are like more relevant in this case. So we start here. I'm representing in the bottom right corner of your screen the action potential of a pacemaker cell. So basically, that's the shape the action potential has. And as I told you, this is an autoregenerative action potential. So it doesn't have a resting membrane potential, but it has this phase that is called the diastolic depolarization. So the diastolic depolarization is what confers automaticity to the pacemaker cells. It's a slow depolarization that brings the membrane potential close to the threshold and then allows the autoregeneration of the action potential. So we're going to start there. And the main two components identified so far contributing to the early diastolic depolarization are precisely the HCM4 channel that I mentioned before. That's why we use it as a marker for pacemaker cells. And also a spontaneous release of calcium through the ryanodine receptor from the sarcoplasmic reticulum. Then this phase is followed by a late diastolic depolarization, which mainly is carried by the opening of two different types of voltage-gated calcium channels. T-type that are expressed in a very low amount in the pacemaker cells but are still present, and L-type calcium channels, specifically the CAV 1.3 channel that activates and more hyperpolarized voltages than the 1.2. There is also evidence showing that there is an increase in the activity of the sodium-calcium exchanger that contributes to this change in the membrane potential and to the activation of the voltage-gated channels. So once we reach the threshold, the action potential is fired, and different to other cells in the heart, here the action potential is mainly carried by calcium. So this is a calcium action potential, and the two main channels contributing to the upstroke of the action potential are the CAV 1.2 and the CAV 1.3, both members of the L-type calcium channels. And these are the channels that I'm going to be talking about the rest of the talk, because those are my favorite channels. So, and then the action potential is repolarized. The membrane potential is repolarized again through the opening of multiple potassium channels. So potassium channel lovers, don't be mad at me because I abbreviated everything in just a single yellow channel here, but obviously there are many kind of potassium channels that are important for this repolarization. So here we go down and the cycle starts again immediately because that repolarization opens the HCM4 channels that are open at hyperpolarized voltages. So this is the way the action potential is regenerated. So if we look into these action potentials in the real animal, and so what you see here is an spontaneous trace. So here we're doing a patch clamp in the current clamp mode from isolated cells. So that in that upper right corner, you see how a pacemaker cell looks like under the patch scope. And so all of those white peaks that you see there are the spontaneous action potentials fired by the pacemaker cells. This is how the spontaneous firing or intrinsic firing rate looks in a young animal. And when you compare with a cell that was isolated from an old mouse, so as you can see there, we work with young adults, so we consider young animals between 4 and 6 months old. And for old mice, we work mainly with animals that are over 24 months. For most of the data that I'm presenting today, we have a group between 28 and 32 months old. That is where we see stronger differences in terms of the L-type calcium channel. So you can see there in the orange trace that first thing probably that you notice is that there are pauses between the firing. So this is an arrhythmic firing, and also it's a bradycardic firing, what means that it beats lower than what the young is beating. So from these action potentials, we can go and do a lot of measurements, as you know, and we can characterize different parts of the action potential, upstroke, duration, also very important for the function of the pacemaker. As you can imagine, we can characterize the early diastolic depolarization duration and the slope of these phases that tell us a lot about what's happening here in terms of the conductances that are being activated. And obviously, the time that passes between each peak, that is what determines the firing rate. So if we can quantify all that, and what I'm summarizing here in this plot is the fold change observed when you compare the old pacemaker cells relative to the young animals. And so everything that we see here highlighted in red are the parameters that we found significantly different. And so first thing, as we expected, the action potential firing rate is lower for old mouse compared to the young. Diastolic depolarization duration is longer. And I don't know if you see that arrow there. Okay. And when we characterize, as I said, the slope of that early and late diastolic phases, we find that these are slower. So it takes more time for the cell to depolarize to the point in which it can reach the action potential and fire again. So if we focus, like if we zoom in that region, talking about the slopes, this is how it looks. So again, we are just seeing into this region here. And in white, we have the diastolic depolarization for the young animal, orange is the old, and then green shows the slope for the early diastolic and blue for the late diastolic. And you see how in the quantifications of these parameters, we see a significant reduction in both phases. I'm sorry. So the fact that we see a slowdown of the diastolic depolarization suggests that aging is impairing the conductances that contribute to this phase of the action potential. So what is known about that, mainly the group of Kathy Proenza is the group that has contributed more to our understanding of how aging affects the cardiac pacemaker currents and action potential. And they have a very big amount of evidence showing that aging impairs the function of the HCM4 channel. So as you can see here, they observe a reduction in the current density in old animals and also achieve in the activation curve of the HCM4 channels. Basically, what that means is that in old pacemaker cells, it's more difficult for the HCM4 channel to open and then contribute to the diastolic depolarization. The question that we had here is, what about the channels that contribute to the late diastolic depolarization? And specifically, what about the T-type and L-type voltage-gated calcium channels? So we decided to take pacemaker cells from young and old animals and study the L-type calcium current. So what we are seeing here is patch clamp in the voltage clamp mode. And here we are stimulating the cells with a square pulse going from -70 to -20 millivolts to open the L-type calcium channels. And what you see here is how the calcium channel currents look. So this is a young and an old representative traces. And this is the total calcium current. So we then apply nifedipine, that is a specific blocker for L-type calcium channels. And therefore, the nifedipine resistant current is what we are going to quantify as the T-type component here. As you can see, is very small, as I said before. And then subtracting that from the total calcium current, we can determine which is the L-type calcium component. So quantification of this data, what shows is that there is no difference in the current density for the T type component of the calcium current, and there is a significant reduction in the L-type calcium component. We also quantify the percentage of the total current that was carried by both types of channels, and we didn't see any shift in which channel is used preferentially in one age or the other. We corroborated that there was, in the case of L-type calcium channels, no change in the voltage dependence of the channels, as we know happens for the HTM4. Is not the case for the L-type. This was just a reduction in the total current. At the single channel level, so we wanted to see how this looked using the single channel recordings, and corroborating what we observed at the global level of the calcium current. We observed a decrease in the open probability of the channel. So what you are seeing here are 4 representative traces from different cells in the single channel cell attached configuration. And you can see each dotted line that you see here is a quantile level for openings. Opposite to many of the single channel experiments that you see, where people is more interested to study dynamics and what is happening with one single channel, we are more interested in seeing multiple events, because many of the analysis that we do in our lab also takes into account the cooperative opening of calcium channels. So that's why probably these recordings look a little bit different from what you are used to see. And actually, this is what you normally get when you do single channel recording. The more difficult thing is to get single ion channels than many, because like many of them are together. So in terms of what we observe, so we observe in the white traces young, all traces again orange. And you can see there that there is a lot of activity, many channels opening at the same time, closing at the same time, and in general, more activity compared to the old cells, in which most of the patches have like individual channels, or probably 2 channels opening, and with a very low open probability. So here, my postdoc, when they came with the idea of asking if we would be able to change the firing rate just by increasing the open probability of the calcium channel. And so one challenge, I wouldn't say disadvantage, but one challenge of working with some primary cells like pacemaker cells is that you are very limited in the amount of tools that you can use. So when we work in a heterologous system, we normally use very fancy optogenetic tools to play with the channels and make them open and play with their activity. But here we were more constrained to pharmacological tools. So we decided to use a BEI-K, that is a drug that is known for increasing the open time of L-type calcium channels. And what we did was recording first in control solution. So what you are seeing here again is a representative trace of spontaneous action potentials for young and for old animals. And then we applied BEI-K 500 nanomolar into the solution. And this is what we observed. So there was a very dramatic increase in the firing of the old cells. And then quantifying this, you see that the significant reduction that we have in firing rate in control conditions is reverted when we apply BEI-K. That means when we make the channels open more. And that supports our hypothesis that the reduction in the global calcium current and this decrease in the open probability of the channels is one of the mechanisms responsible for the age-associated slowdown of the pacemaker. We also did, like all the analysis of the action potential, but I wanted to show you again what is the change at the level of the diastolic depolarization that at the end, it was what originated the question of what aging is doing to the conductances that are important for that diastolic depolarization. And what you are seeing here is, again, control conditions, young, white, orange is old, and we see the difference in the slope. And after we apply K, there is a change in the slope and now an acceleration. So we have a recovery in the duration of the diastolic depolarization and also in the late and early. That difference that change that we observed in the early diastolic depolarization is surprising, and I mean it needs further experiments to corroborate that, but it suggests that this CAV 1.3 channel could be contributing even at those very negative potentials into that depolarization. Okay, so, but what is the mechanism behind the reduction of the calcium current in all pacemaker cells? So here we decided to go and study many things that we know are important for the L-type calcium channel function, and one of them is how they arrange at the plasma membrane. So pacemaker cells probably are not very well characterized in terms of structure as the cardiomyocytes are, but this is a nice hypothesis paper that Alexei Glukov, who also works with the pacemaker cells, published a few years ago, in which he has this nice diagram that I keep using because I like it a lot. And I think I even made this version in a black background, so I have to send this to Alexei if you want to use that. But basically, what Alexei shows here is that even though these cells look very different from ventricular cardiomyocytes, as I told you, so you can see that they don't have T-tubules, that is one of the main characteristics of cardiomyocytes, but they rely on this extensive network of caveoli for organizing all these ion channels that we know are important for the pacemaker function. Here, like many people in the field, hypothesize that the location of these channels here in this caveoli is important for that crosstalk between channels to achieve the activation and keep contributing and amplify the diastolic depolarization to reach the threshold, and also for the crosstalk between the channels and the receptors that sense the noradrenaline that I told you that is important for accelerating the heart rate. Okay, in this diagram, Alexei showed more like CAV 1.2 channels here, and he showed 1.3 more like in other structure and other cells that kind of are at the periphery of the pacemaker. But we wanted to test the idea that aging is messing up with this whole structure and organization. So we went to first immunodetect caveoli in pacemaker cells. So what you are seeing there on the screen are representative images of pacemaker cells isolated from young and old animals, and immunostained, again, caveoli. So as you can see, the young cells do show a really nice continuous labeling across the membrane, while the old cells look more fragmented. So we went to use expansion microscopy to try to look in more detail at these alterations in the continuity of the caveolar network. And what we observed there, so what you're looking here in the screen, are two planes, one taken at the middle of the cell, and this is an inverted fluorescence image. So what you see there in black is the fluorescence for the antibody detecting caveolin. And again, you see how for the young, there is a more continuous network compared to the old. And this is how the staining looks at the footprint of the cell. And we saw a more discontinuous and less dense caveolin network. So here we quantified the length of these caveolar areas and the total area occupied for caveoli, and we concluded that there was indeed a decrease in the caveolar network. So to test the idea that aging was disrupting the localization of L-type calcium channels inside this caveoli, we use a approach called proximity ligation assay that probably people now are very familiar with it, but I still have this slide, so I will go through this. But basically what PLA is that when you want to determine if two proteins are close to each other, closer than 40 nanometers, or if you want to test if there is any kind of treatment that disrupts the proximity between the proteins that you are interested in, then what you do is to use primary antibodies directed to these targets that you are interested, and then use secondary antibodies that are tagged with these DNA or oligo probes that if your proteins happen to be closer than 40 nanometers, then they will undergo a ligation reaction, and then you can amplify. So basically it's like doing a PCR on top of your proteins of interest, and every time you do an amplification, there is an incorporation of a probe that is tagged with a fluorophore. So what you ended up seeing is something that looks like a bright spot on top of the proteins that are close to each other. So we use this approach to test proximity between the CAV 1.3 and the caveolin 3 protein. And again, this is an inverted image of a fluorescent image. So what you are seeing there in black are those puncta, fluorescent puncta that we get from the PLA. So this is how it looks for young animal. And this is what we observed for the old animals. So there was a striking reduction in the number of puncta or places where the CAV 1.3 channels were close to caveolin. So we have reported before, and I'm not going to present any data about that, but we have reported that this proximity between calcium channels, the fact that they organize in microdomains that allow them to be together, is a fact that is important for the function of the L-type calcium channel. So we have shown using super resolution microscopy, storm microscopy, that ion channels organize in these clusters that are important for the function. So for people that are not familiar with the use of super resolution to study ion channels, I strongly recommend you get into that world. It's very exciting when you use super resolution microscopy and change your whole perception that ion channels don't distribute in an homogeneous way in the pacemaker in the plasma membrane, but they actually form clusters. So this is an image that you would get from any of the regular microscopes that probably you have in the lab. But if you use super resolution microscopy, then it changes completely the view that you get of how ion channels look like at the membrane. And the more channels we studied, we did this in a diversity of cells. During my postdoc in Fernando Santana lab, they have reported a lot of data showing that, in fact, ion channels organize in these large clusters, and that organization is very important for the modulation of the channel. And so we studied also how aging was messing up with this clustering. So this is just a portion of a pacemaker cell, because this is, we do these experiments with high magnification objectives. So what you are seeing there is just the footprint of the cell, and all those dots that you see there are CAV 1.2 channels that have been labeled immunolabeled and detected with a fluorophore antibody. And so this distribution was similar to what we have observed in other cells. And when we went to look into all cells, so we saw again a big reduction in the clustering of the pacemaker cells. And here is just the quantification to show that aging causes a decrease in the cluster density and also in the cluster mean area for both the 1.2 and the 1.3. And we also have evidence showing that all these disruption of the microdomains where the channels insert and the reduction in the formation of these dense clusters also affect the way these channels are interacting or organizing relative to other proteins. In this case, specifically the one where we are interested is the beta-adrenergic receptors. So as I mentioned at the beginning, there is a parallel decline in maximum heart rate and intrinsic pacemaker rate. But if you put these lines together, you can see that the intrinsic pacemaker rate cannot explain completely the slope that we get in the maximum heart rate. What that suggests is that the slowdown of the pacemaker has two components. One that depends on something intrinsic of the pacemaker rate, and it has a small component that is due to a reduction in the way the cells are responding to the adrenergic stimulation. So if we go back to our diagram, what you see here is that all these channels, and in our case, we're interested in the CAV 1.2 and 1.3, are regulated by noradrenaline to the binding and activation of the beta-adrenergic signaling pathway, in which there is an increase in cyclic AMP, activation of PKA, and phosphorylation of many targets. So this is the mechanism by which the heart is accelerated. So we used PLA to evaluate how aging is affecting the proximity of these channels with the beta-adrenergic receptors. And what we observed is pacemaker cells express both beta-1 and beta-2 adrenergic receptors. And we observed that aging affects specifically the interaction of both L-type calcium channels with the beta-2 adrenergic receptor. So this is what I'm showing here. These are representative PLA images for the 1.2 and the 1.3 channel, testing their proximity to the beta-2. This is a young cells from young animals, and these are cells from old animals. And you can see here in the quantification how there is a reduction in the 1.2 and a very dramatic reduction for the interaction with the 1.3 channel. We tested these also for the beta-1 adrenergic receptor. And again, here, these are the representative images for 1.2 and 1.3. And we observed an increase for the association with the 1.2 and a reduction for the 1.3, showing that these channels, as it has been seen for ventricular cardiomyocytes, might have different roles and preferences for the association between these two receptors. At the functional level, we also saw a difference. So we use an approach in which we recorded a time course for the calcium current. So what you are seeing here is just the fold change in the calcium current amplitude against time. So we have a baseline, then we apply formoterol, that is an agonist for the beta-2 adrenergic receptor. The orange trace is showing the response in old cells, and the black one is showing the response for young cells. And then we activated the beta-1 with an unspecific agonist, but with the idea that the beta-2 was already activated by formoterol. And we evaluated the amplitude of that fold change. Then we wash the agonist, and then at the end apply nifedipine to show, to calculate what percentage of that current was carried by the L-type calcium channel that we know is most. So the quantification of that experiment, what it shows is that there is a significant reduction, again, in the facilitation that is exerted by the activation of the beta-2 adrenergic receptor, and no change for the beta-1, showing again that aging affects specifically. Okay, and so I'm gonna jump to this slide for concluding. So some of the things I show today is how aging is affecting ion channel function in different ways. We think definitely there is a big component that we are studying right now, and is how aging can affect probably trafficking and insertion of the ion channels at the plasma membrane to account for the reduction in density and clustering that we are seeing at the plasma membrane. We think one of the mechanisms could be the disruption of these caveolar domains, where the channels probably are delivered and stay there. So we think this disruption of the caveolar network might be one of the reasons, because we have less channels at the plasma membrane. And we show that how that affects the interaction of the channels with the beta adrenergic receptor. And I just wanted to put a brief teaser for students and people that are interested in looking for postdoctoral positions soon. I'm about to open a couple of positions in my lab. We do many things apart from my aging research that I showed you at the beginning, but one of the most exciting things that we are jumping into right now is to study the pacemaker of the most extreme endotherm in the animal kingdom, the hummingbird. The hummingbird reaches heart rates of about 1,200 beats per minute, and this is opening a big window for discovery for us. We're very excited about the things that we're getting with this project, and I hope soon to put together a seminar to start showing people what we have got. And so if you are interested, you know how to reach me. And I just wanted to end by thanking again the people from my lab that make all this possible and are always on board for the crazy ideas that I have every day for experiments. And obviously the people that support my funding, the NIH and also the HHM. And I'm happy to take questions.
Okay, thank you very much, Claudia. This was really great. We already have a few questions in the chat, so I'll just start with those. So Shin-Fluid Lim says, great talk. I might have missed this, but was there a difference in resting membrane potential? I'm guessing old and young pacemaker cells.
Yeah, you didn't miss it. I pointed out at the beginning. So there is not such a thing as resting membrane potential in these cells, because as soon as they depolarize, so we have something that is called the maximum diastolic depolarization. That would be the closest that we can get, and there we didn't see any significant difference at that level.
Okay, and then the next question is from Damien Bell. Lovely single channel recordings. The reduced open probability seems to be a cause of reduced calcium currents in aged pacemaker cells. Is it possible to discriminate between CAV 1.3 and 1.2 for this age-related reduction in open probability?
Oh, I love that. I love that question, because you know, people working with voltage gated calcium channels, we have always had that problem and that dream of being able to separate them. When we started this project, we suggested using the CAV 1.3 knockout and also the transgenic that is resistant for the hydropyridine inhibition for the 1.2. But quickly after I started the project, I realized that was not a good idea, since I had to age mice for 2 years to be able to do that. So I told the people from the NIH, no, we're not gonna do that. So I changed directions with that. But luckily, there was a paper published, I think this year, and I don't remember the group. But if you look for it in the web, you will find it easy. Is the characterization of a toxin that comes from the black mamba snake. And the data looks very strong. It shows that it has a preference for blocking CAB 1.2 over CAB 1.3. And we are testing it right now. So we are doing the characterization first in just a heterologous system to kind of play with the ratios and make sure it works in our hands. And the idea is to use that to try to see, because we saw a difference between the 1.3 and the 1.2 at different levels, the way they associate with the receptors, the decrease we got in terms of the localization in the caveolin. I didn't show that data, but we also saw big difference between how they are associated to ACAP, that is a scaffold protein important for the beta-adrenergic. So I think we have enough evidence to think that aging is affecting them in a different way. And so I hope that toxin will give us an answer.
Okay. So the next question is again from Shindruelin. Have you looked into changes in channels involved in electrical transmission, like gap junction, function expression with aging, or channels involved in calcium waves like IP3 receptors and ryanodine receptors?
There is, I think, large evidence from the Lakata group showing mainly changes what happens at the level of the sarcoplasmic reticulum. We haven't explored other channels yet because I think the L-type calcium always keep us really busy. But now I see Jorge is here, so I am trying to convince Jorge to collaborate with me and see what's going on with the connections. You know, with I have. Let me see if I have a slide here that I can share. One thing that we see in the pacemaker, so what you are seeing there is how aging messes up with the whole architecture of the pacemaker. So this is a cleared pacemaker comparing young and old. So the young is the nice structure that I showed you at the beginning. This is how it looks in the old. So there is a lot of disorganization, probably a lot of fibrosis there. So for sure, connectivity and electrical coupling between the cells is being affected, but that's something that we haven't explored at the functional, like recording in the whole explant.
Okay, so the next question is from Tina Borstedt. Thanks so much for the fantastic talk. I have some quite naive questions. Do you know whether someone had a closer look at mitochondria and the mitochondria-associated membrane systems in this context? Since the process needs quite some ATP and cyclic AMP and the calcium transport via SERCA, etc., did someone had a look at changes in ATP levels in this context? And also, like there's a question regarding the clustering. Can it also, at least in part and indirect effect due to changes in total local calcium concentration? So could it be affected by the changes in local calcium concentration, the clustering changes that you observed?
Mm-hmm. Okay, so the first question about the mitochondria, I think is very interesting, and I'm almost sure that if we look into that, we will find a difference. Something that I didn't mention is that the head of the pacemaker, that is where the kind of what people call the true pacemaker cells, or the lead pacemaker resides, are the cells that are more active and that are the ones generating driving the firing. So once they fire, that signal is propagated through the body and then it exits to the atrium. So there are different cell types inside the pacemaker, and there are at least 3 different morphological types. So I think if we characterize that, we should expect to see also heterogeneous activity in terms of mitochondrial activity. And we have now a collaboration with Dr. Amraya Sweetwine, that is an expert in mitochondria, and we are trying to pursue that question of seeing if we are able to dissect different parts of the pacemaker and test mitochondrial activity there. As I said, one of the challenges of working with the pacemaker is also that it's a very small tissue. So the amount of things that I can get from it, it's challenging. That has kind of slowed down a lot of the techniques that I have wanted to incorporate in the pacemaker, mainly because the amount of cells and tissue that we can isolate for all these assays is limited, or we need to pull many animals to do that. In terms of the calcium microdomains, that's very unexplored in these cells, because even though there is evidence showing that crosstalk between the sarcoplasmic reticulum is so important for the activation of what we call the membrane clock, that it would be all the ion channels residing at the plasma membrane, there is not a clear consensus right now on how that happens. We don't have T-tubules there. There is not a big characterization on how the sarcoplasmic reticulum is organized in these cells. There is a couple of papers showing that these cells, in fact, have some prototubules that we think, and we are testing if they have a functional role in regulating those calcium microdomains that might be important. But I think, because we don't know yet how is that happening, like in normal young animals, we're still far from seeing what's going on in terms of alterations with age.
Okay. So we have lots of questions in the chat. That's so great to see. I'll just read out. We are now it's at the end of the time, but I'll read out 3 more just because they're so interesting. And also Tina has a follow-up questions. But Tina, if you're interested, I would encourage you to maybe email Claudia with that because we won't have time. But Hartan Tun asks, are the cells in pacemaker heterogeneous? Do you see difference in functional expression of calcium channel subtypes between cells?
Yes, so they are heterogeneous, as I mentioned. There are 3 phenotypes. So we are using, in one of our projects, we are using patch-seq. Like, if you think patching pacemaker cells is difficult, try to collect them to do RNA sequencing after you are done with the recording. So that has been a very exciting and challenging project. But the idea behind that project is that we want to identify, like if or determine if they are really functionally different. They look different. We know they distribute different in the pacemaker, but people don't know exactly, how different functionally they are. There are some reports showing that one of the main differences between these cells is that they have different densities of calcium channels, as you say, and also the density of the HCM4. So the belief is like, the closer you are to the center of the pacemaker, you will have these cells that have more calcium channels and more HCM4. And the more you move out of that region, then you start having a gradient of decrease in the expression of HCM4, and then some expression of the the sodium 1.5 channel.
Okay. Thank you. So the next question is from David Colhoun with self-reported interest in aging, because he's 87. So he asks, the variance seems often to be very big in the young and smaller in the old mice. In some cases, the young points look almost bimodal, with one peak very similar to the old. So what do you think about that? Did you notice that?
I think I don't know if they are talking about the density data, probably. Yes, and I think, like intrinsic, and I have probably to do a disclaimer here. All of our recordings are performed at room temperature, and that obviously affects that, like the intrinsic pacemaker rate that we are observing there. But as I said, like pacemaker cells are heterogeneous. That's something that has been, like extensively reported in the field. There are some pacemaker cells even that you take them from young animals and they don't fire spontaneously. And so there are many hypotheses on, like how these cells work now as an oscillator and integrate, who kind of trigger the other and drive the action potential, even though they all have the machinery to keep beating once they get started. So we haven't, I haven't gone deeper into exploring the different populations electrically because it will require, I think, many more cells to reach a conclusion on different populations there.
Okay. So I think Damien Bell just had a comment on the black mamba toxin. You can see that. And Hader Cruz says their group is studying heart electrophysiology in bats, which is very interesting. They jumped from 40 BPM to an amazing 1000. So some very interesting phenomena out there. Okay.
Yeah, there is a thank you, Damian, for the link. Yeah, I was very excited when I saw that. I bought it the same day. I read the paper. I need that on my shelf.
Okay.
Yelena, can I ask one quick question?
Yeah, sure. Okay.
Claudia, I really enjoyed the talk and I learned a lot. I'm wondering about where these changes come from. Is it intrinsic to the nodal cells themselves, or is it some response to systemic factors? And has anyone done one of these creepy parabiosis experiments and seen whether there's any rejuvenation of the old heart if you plumb in a young mouse?
I think from this famous study in which they kind of killed a bunch of senescent cells in the mouth and they would able to rejuvenate. I think one of the parameters that they measured was cardiac performance and heart rate, and I think they saw a change there. That we haven't. I mean, if there are many studies using, for example, rapamycin that show an improving in cardiac performance, but none of those studies look directly to the cardiac pacemaker. There are just a few groups that work on that, and I think not many in aging. So, about the first question, I think it's a mix of both. So definitely there should be some senescent processes happening in the pacemaker cells, and as it was mentioned before, these are cells that are super active metabolically. So I think I would expect, or I have a hypothesis that I'm trying to test, that those cells in the head that are the leading, the cells that fire faster are the ones that are more affected by those changes. Probably a lot of oxidative stress and like decline in mitochondrial function. We are thinking that then when you lose those fast cells, others have to take over, and then luckily the heart has a way to keep it beating because we need it. And so other cells can, can function inside the pacemaker as the lead, but then probably are cells that intrinsically are slower than the other. But, that question of who's driving who, because we know the sympathetic nervous system also changes with age, and there is an increase in the production of noradrenaline. So if we have more noradrenaline spilling over the heart, or even, if you think, now we live in these times where we are all stressed with the adrenaline at the top all the time. So for sure that probably will have a big pressure on how much beta receptor is being expressed, how channels are being regulated, how they are being put together. So I think those are interesting questions that we will have to keep asking.
Great, thank you.
Maybe even t-test?
Yes, I do.
David, I think you can. Greg, can people
Yeah,
just
so I saw a couple of comments about the. If you want to contact me for more questions and for collaborations, I'm always open for collaboration. We have like tons of ideas that we will be really willing to test.
Yes, one problem is likely to be that these very unequal variances will invalidate a t-test. But all these p-values coming from t-tests.
Oh, sorry. I was trying to move the slide. So you were referring to the data for what exactly?
The scatter for the young was often very different from the scatter for the old, and that would invalidate a t-test. I wondered if you
But for,
values
I mean, I showed it several different scatter plots, so I
Yes,
don't know which one you are referring.
well, in very many of them, the scatter was very different. So a t-test assumes equal variances in the two groups. But did the p-values come from t-tests or from some other test?
For some, yeah, for some we did t-test. And for, yeah, I don't have all the data here, but
Yes.
yeah, I mean, we can totally discuss that, and we could run some tests to show the difference in the variance and make sure that is included in the paper when we submit it.
Yes, there's a thing called the
I
Welch,
appreciate.
which allows unequal variances. If you believe in p-values, I don't believe in p-values anyway, but that's a different question.
Okay, yeah, I will take that into account. Thank you so much, Dave.
Thank you.
Okay, I think now it's time to finish. There might be another collaboration emerging based on the comments. So thank you so much, Claudia. This was very interesting.
Oh, no. Thank you so much for the invitation.
Yeah, good luck with the future research, and thank you everybody for coming. Our June seminar is Sampurna Chakrabarti, who is in Gary Lowen's lab, and they just had a paper about Elkin-1, which is a sort of relatively novel mechanism, sensitive ion channel that mediates normal touch sensation in mice. Okay, thank you very much. Bye.